at the Duke Molecular Physiology Institute, and we're really lucky today to have Dr. Stan Hazen giving DCRI a research conference. Stan has really stood up to me as an exemplar of where I hope to go in my research, and am realizing through his talks that I will never reach, but he's really done phenomenal work. Stan has an interesting background. He did a PhD in biophysical chemistry and molecular biology at Washington University prior to doing his internal medicine residency and endocrinology fellowship, and then was a Howard Hughes Medical Institute postdoctoral fellow. He then came onto faculty at Cleveland Clinic, where he quickly rose in the ranks and has several titles. He is the Jan Bleeksma Chair in Vascular Cell Biology and Atherosclerosis. He's the Leonard Krieger Chair in Preventive Cardiology and actually runs a robust preventive cardiology clinic, so sort of a true exemplar of a physician scientist. He's Department Chair of Cellular and Molecular Medicine at the Cleveland Clinic and within the Lerner Research Institute. Stan's research interests have focused around cholesterol, fundamental mechanisms of cholesterol and HDL, and mechanisms of atherosclerosis. But what you'll see from today's talk is he's really a world leader in translational research, really taking evolving molecular technologies, including metabolomics and genomics, applying them to human populations for discovery science, and then taking those discoveries and really digging into them at a fundamental level to understand their biological implications, and not only that, potentially develop new therapeutic targets to prevent atherosclerosis, which is sort of his ultimate goal. He's really assembled an impressive group across a wide breadth of disciplines, and he himself has an amazing expertise across metabolomics, genomics, quantitative sciences, biochemistry, and metabolism, and is really, again, I think, an amazing example of what we can do in the sort of big science world. He holds several patents, some very interesting patents, several of which are licensed. He has over 330 publications, including New England Journal, Nature Medicine, and Cell. So it's really an honor to have Stan present today, and the title of his talk is Gut Microbes, TMAO, and Cardiorenal Disease. So thanks, Stan.
Thank you very much. So, did you guys need to see these two things? No, then I'm gonna. Here we are. So this is a topic that we've been working on in the lab for about 10 years now. Our first paper came out in 2011, and we've kind of been moving the field forward in terms of understanding how gut microbes are linked to cardiovascular disease through this one particular pathway I'm going to talk about called the TMAO pathway. And what I hope to share with you in the first part is for the most part, kind of review of the introduction of how we discovered this pathway and its link to cardiovascular disease, and how we took it into animal model studies to show that it wasn't just associative but causative. And then actually the majority of the rest of the talk is things that have come out either in the last year or are in press, and that's new connections between this pathway and the development of thrombotic events, in particular in modulating platelet function. So I have every possible conflict of interest you can imagine, but all of the work I'm going to describe for you has been funded by the NIH. All right, so this is a scheme of the overall pathway that I'm going to describe, and that is that when we eat foods that are abundant in cholesterol, and for example, there are other nutrients inside these foods that have a trimethylamine moiety. Oops, I'm going backwards the wrong way. So what I mean by that is phosphatidylcholine, for example, the major building block of cell membranes, is made, it's lecithin. And so that's the major building block of what houses either not just a cell membrane, but also vesicles that house neutral lipids, so fatty triglycerides and cholesterol ester. They're bounded by phospholipids. So you cannot have a diet high in fatty acids or fat unless you're drinking oils. If you're drinking tissue, if you're talking about tissue, if you have cholesterol and fat, you also have phosphatidylcholine. So if you're thinking about a diet that's low in phosphatidylcholine, it's a diet that's low in cholesterol and low in fat. But in addition, I'll talk about a little bit briefly about carnitine, which shares this trimethylamine group. And what I'm going to show you is that when we eat these in our gut microbes will actually cleave between that carbon-nitrogen bond and release this thing called trimethylamine, or TMA. This is a gas at body temperatures. It smells like rotting fish. It's what gives rotting fish or putrefaction. That's the smell of putrid. It's very potent, and believe me, I told the people last night, it will evacuate an entire floor in a building if it's inappropriately opened and not in a hood, a canister. But in any event, TMA in our gut, through the portal blood, everything we absorb through our intestines goes directly to the liver through the portal blood. So anything that gut microbes make delivers to our body through the portal blood. And then in the liver, there's a cluster of enzymes called flavin monooxygenases. These are like cytochrome P450s. They're involved in metabolism of intermediate metabolites. And so like barbiturates, for example, are metabolized by the FMOs. But TMA gets converted into trimethylamine and oxide. So this alcohol group gets stuck on. Now it's no longer a gas. It's a quaternary amine, and it actually stays in solution, and it's excreted in the urine. So it's thought of as a nitrogenous waste product. And what I'm going to show you is that if you eat these precursors, you have higher levels of these in your plasma, but you also have higher risk for cardiovascular disease. But it's only in the setting of a higher TMAO. And what we found is that in animal models, if we feed the precursors, they will make TMA and TMAO only if they have intact gut flora. If you do it in a germ-free mouse or a mouse that you suppress the gut microbes with antibiotics, they don't make those metabolites, and then they also don't get accelerated diet-induced atherosclerosis. So a Western diet is rich in the dietary precursors that make TMAO, and when you get off a Western diet, you suppress, and as I'll show you at the end, you can lower TMAO levels in the plasma. So that's actually the pathway, and let me show you some of the data that argues that this pathway is relevant before I kind of show you the new data. So, I should mention that when we think about our physiology here, it's really, we're talking about the confluence between two sets of organisms, the microbes and the host. So it's a meta-organismal pathway. And we should really be thinking about our physiology in a more global sense this way, because gut microbes play a huge role in our metabolism. There was just a recent paper that came out in PNAS just in the last 2 weeks that talks about fully a third of the metabolites that are easily detectable in plasma are derived or attributable to coming in one way or another via gut microbes when you compare germ-free animals versus conventional animals. So it's a huge component of our physiology. And as an example, there are some vitamins that we cannot make, but we only get them via bioactivation by the gut microbes. Now, our discovery of this pathway began with a study called GeneBank. It's like your CathGen. We collected samples from subjects in the cath lab as well as in preventive cardiology who are undergoing cardiac CT, and we then followed them over time to see who went on and developed heart attack, stroke, or death. So we have over 10,000 subject samples, and we took plasma from a small group of subjects initially, 50 subjects, who we knew went on to develop heart attack, stroke, or death in the ensuing 3-year period after collection of the sample. And we picked an age and gender matched control group. And in each person's plasma, we injected it onto the mass spectrometer. So after the HPLC, as it's coming off, we literally collected full scan mass spectra every few seconds. And so we analyzed what worked out to be almost 6,000 compounds, not knowing what they were other than their retention time and essentially their molecular weight. And we did that in a learning set, and then we did that in a subsequent validation cohort. And then after adjusting for multiple sampling and Bonferroni adjustment, et cetera, we came up with 18 compounds that met the adjusted p-value for significance as being associated with future risk of cardiovascular disease. Now, what were those compounds? It turns out we had no idea. So, but if you look at them on a plot here where on the x-axis is something called MZ, mass to charge ratio. Think of that as the molecular weight of the compound. This is just a narrow window of some of the compounds. On the y-axis is the odds ratio for cardiovascular disease, or actually for MACE, for 3-year MACE. And you'll notice, so it's a forest plot on its side. And these 95% confidence intervals are fairly broad, but if you look at where the odds ratio are, it's pretty high up there, like, you know, in the 5 to 10-fold range. So these are big. By comparison, LDL cholesterol, this is just in the validation cohort, did not reach significance in our group. So then you're saying, well, what are these? And like, what is that one way up here? Well, that ended up being citrulline. But you'll notice the 95% confidence interval comes down here to about 5. We had just previously done a metabolomic survey around arginine metabolism and nitric oxide metabolism and showed that citrulline was a very strong predictor of cardiovascular risk with an odds ratio of about 4.8. So it was just below the 95% confidence interval. So we've identified all of these compounds, and I'll tell you 3 out of the 18 shown here can only be made by bacteria. Now, the 3 that we were most interested in were the ones shown in black. We started with these 3 because they were correlated with each other very highly, and so it made us think that maybe they're involved in some kind of pathway. And so, because they had such a high correlation with each other, also it's easier to figure out the structure of something that's small compared to something that's large. We started focusing on these 3, and the net result is those 3 were linked to phosphatidylcholine metabolism. They ended up being, the lowest molecular weight one was this thing called, I'm just going to point like this, was trimethylamine N oxide. I should point out, as I was talking at lunch, this has the exact same elemental composition as glycine. And in fact, our instrument, which had the first metabolomic software package made by AB Science, was calling it glycine. And it's like, no, it can't be glycine. That makes no sense that glycine would predict cardiac risk. And it ended up not being glycine. And it was, now that it's been trained better, it does reproducibly predict it as trimethylamine N oxide. But the other 2 compounds were choline and betaine. Betaine is just simply the oxidation product of choline, where the alcohol is a carboxylic acid. And so all 3 of these compounds are linked to phosphatidylcholine metabolism. Now I'm going to just take a step back and say, well, wait a minute. You know, you probably are thinking, phosphatidylcholine metabolism, I've never heard of these. Why are these listed? Well, when we eat a fatty acid, or phospholipid, lipases are from the pancreas are emptied at the, you know, into the duodenum, and so you are into the, and so you end up having pancreatic lipase cleave off the fatty acids off of the phospholipid, and then glycerophosphocholene is what, for the most part, starts to work its way down further in the intestine, and then also it can get cleaved to generate either phosphorylcholine or phosphocholine or choline, and so that combination of stuff gets absorbed in the small bowel, but a significant portion does not. And depending on how much you eat, you may or may not saturate your choline transporters in the small bowel. And so when it reaches the colon, it's a bioreactor, and that's where the bacteria are that do most of this biology, and will cleave the choline head group off and release the TMA and the TMAO. And we can tell that by literally segmenting the intestines and doing just an assay by adding isotope-labeled choline to the homogenate of these different sections and seeing where does the choline get converted into TMA, for example. Now, so early studies identified these 3, and so we developed very specific assays for quantifying all 3. And then we, in a separate non-overlapping group of over 1,800 subjects, we looked at the levels and just focus on the TMAO in the middle. You'll note that this is like between the 95, the 5th and the 95th percentile of our cohort. So the levels can get quite high. What you'll see, though, is that it's very steep. Look at how deep the odds ratio is. So the higher the TMAO level, but the higher any of these levels, the higher the likelihood of cardiovascular disease. And what we found, though, is that in subsequent studies, I'm not going to show you here, though, but if you adjust for TMAO, the predictive value of choline and betaine, they fall out. It's all related to TMAO. And of course, choline and betaine are linked to how much of choline and betaine you ingest. And so the bottom line is we think that this replicated our early data of identifying an analyte that tracked with cardiovascular risk. Now, I'm just going to show a lot of kind of clinical studies data on TMAO in different cohorts, but here's looking at it in a different 1,000 subjects who had undergone angiography and just very simple scoring of 0, 1, 2, 3 vessel disease. More recently, we've done syntax score 1, syntax 2, and you see a direct relationship between TMAO levels and atherosclerotic burden in a subject. In this particular study, over 4,000 subjects who are in our gene bank cohort, non-overlapping with the previous ones, we measured TMAO and then asked how does this track with future risk of heart attack, stroke, or death, or the composite of major adverse cardiac events over a 3-year period. And so the first 1,000 subjects, or first quartile, was our index reference group, and a fourth quartile, so the top 1,000 subjects, they're about a 4-fold increased risk of major adverse cardiac event, MI, stroke, or death, over the ensuing 3-year period. That's the 95% confidence interval. And they had overall poor survival in a dose-dependent fashion, the higher the level of TMAO. Oops. So what's interesting about this is that the relationship between TMAO and cardiovascular risk is in large part independent of traditional risk factors. So this is looking at 3-year risk of major adverse cardiac event, MI, stroke, or death, stratified by multiple different Framingham and other risk factors. And I'll just point out a couple things. First of all, for example, LDL cholesterol level, even with a level less than 70, a high TMAO is predicting increased risk. So this kind of barely, I don't know if you guys can see the pointer. So that, to me, argues that, okay, if this is causal, that perhaps there's an extra pathway here that we might be able to tweak in helping to lower risk even after we've brought someone down to a so-called at goal with their statin therapy. But the key thing here is that the risk between TMAO and major adverse events is in large part independent of the traditional risk factors. Now, early on, we didn't know where TMAO came from, but we had a suspicion that it might come from gut microbes. So to test this, we got germ-free animals. These are born sterilely by cesarean section. They're maintained in microisolators with all sterile autoclave food and water. If you actually look at their feces, the DNA content from bacteria is incredibly low, like 1,000,000-fold lower than a regular animal. There's still some detectable bacteria because food itself has bacteria, even if it's autoclaved, can have bacterial DNA in it, but it's just dead. So taking these germ-free animals, we then gave them, instead of a glucose tolerance test, we gave them a phosphatidylcholine tolerance test. So we gavage them with phosphatidylcholine and then look at the metabolites derived from phosphatidylcholine as they appear in the plasma. And what we saw is that very early on, choline and betaine show up in the plasma, but then no TMAO at all appeared in the plasma. And so in the absence of gut microbes, you didn't make any TMAO. If we then took the animals and we put them in conventional cages so they get colonized quite rapidly, then you give them the phosphatidylcholine gavage, now you make TMAO. And you'll notice the time course, what happens is as the food, the phosphatidylcholine is going down, or when the choline reaches it has to reach the colon before and reach the bacteria that can do the reaction. And so that's where you get this kind of time course. And now with mice, we see a much bigger effect than in humans. I have to say that in humans, after eating a meal, the effect on plasma TMAO is not that big. What is by far the bigger effect is the chronic dietary exposure. And I'll show you data to this effect, where we've taken subjects who are vegan or vegetarian or omnivore and put them on different diets and show what has a bigger effect on TMAO. And it's by far and away chronic exposure that has the biggest effect on the plasma levels. Okay, so that was mice. What about humans? So this is just a pictorial illustration of a mass spec detection off of a HPLC column. So the y-axis here is retention time on the HPLC column. It's not time after ingestion. What I'm showing you in the top is we gave them 2 hard-boiled eggs in a capsule with isotope-labeled phosphatidylcholine, and we were looking for TMAO to appear in the blood that was either natural abundance derived, like from the egg yolk, or the isotope labeled from the capsule. And so in the plasmas, the top 2 panels, the bottom 2 panels are urine. And at the time we looked at, 6 hours following challenge, you could see quite readily that TMAO was there. But then after being placed on a cocktail of poorly absorbed antibiotics, gentamicin, vancomycin, neomycin, metronidazole, ciprofloxacin. This is a cocktail of 5 antibiotics that actually rectal surgeons used to use to sterilize the intestine before rectal surgeries a long time ago. They don't do that anymore. But this was a brief antibiotic cocktail, and we saw that very rapidly. It suppresses the gut bacterial composition, by over 10,000-fold, and now we give the challenge and very little detectable TMAO. Then they go off the antibiotics, come back a couple weeks later, now you give them the hard-boiled eggs in the capsule, and now you see it. So it's essentially now you see it, now you don't. So in humans, when we measure this stuff in plasma, it has an absolute requirement for gut microbes to make this stuff that's at high level in our plasma. And it's kind of amazing. Things at a level this high is coming from gut bacteria, but TMAO is not unique. There are many, many compounds in our circulation that come from gut microbes, and many of them are not only there after you eat. They're actually there all the time. So for example, TMAO, the natural abundance material, if you fast, the level will come down a little bit, but not that much, and then you fast 24, we've gone out to 72 hours. The plasma level stays relatively the same, and that's because our colon is a bioreactor. It's constantly generating metabolites, and we're constantly, I just killed this, oh there we go. We're constantly generating more TMAO or more TMA because we're constantly feeding the bacteria. Our epithelial lining of our gut turns over every 3 days and sloughs off, and so it gets auto-digested. Mucus is constantly being generated, and many of the microbes only survive on mucus as their fuel source, and there's a lot of phosphatidylcholine connected with mucus. And then every time you eat and your gallbladder squeezes, it's not just bile salts that you put into your intestine. By mass, there's more phosphatidylcholine than bile acids in bile fluid. So we emulsify what we eat as a way of digesting the lipids, and so we're constantly feeding the gut microbes phosphatidylcholine. Everyone, whether you're a vegan or an omnivore. And so the microbes, it's a constant generation of this stuff. Now, all of this suggests an association with heart disease and the ability to predict risk, but it doesn't mean it's connected to causing the disease. The studies that make us think that that's involved are this, is one of the first ones, and that is we looked at the ApoE knockout mouse. That's an atherosclerosis-prone animal that develops atherosclerosis spontaneously, and it's hypercholesterolemic, and so on a normal chow, these animals have a high cholesterol. What we did is we supplemented the cholesterol with choline at a level of what you would expect like in a Western diet. So if you just focus on the open bars, those are the animals with intact gut microbes. When we put them on a choline supplemented diet, they get accelerated atherosclerosis. We're measuring atherosclerosis in the aortic root of the animal. So that's the choline animals. And in red, you can see the TMAO levels. So on a high choline diet, there's about a 10-fold increase in this particular diet in the TMAO levels in plasma, but it's well within the range of what we see in humans. If we instead put TMAO into the food, and we happen to get very close with this, we get about a 10-fold increase in TMAO levels in the plasma. You get accelerated atherosclerosis. Now the black bars, those animals had chronic antibiotics in the drinking water, and so it suppressed the gut microbes. And if you look at the plasma TMAO, even though they were on a high choline diet, their TMAO was non-detectable. So you're suppressing the bacteria, and you fail to see the diet-induced enhanced atherosclerosis on the high choline diet. But if you bypass the gut microbes and give them the TMAO in the food, now they get the accelerated atherosclerosis. So data like this argued that TMAO was somehow having a direct effect on atherosclerosis progression in the mouse model. Now, I'm not going to show you, in the interest of time, the data with carnitine so I can get to new material, but if you put carnitine in the diet, in an analogous way, you accelerate atherosclerosis in the animal model, but only if they have intact gut microbes. If you do it in a germ-free animal or you suppress the gut microbes with antibiotics, you fail to see TMAO generation with the carnitine, and you fail to see enhanced atherosclerosis. And so you're asking why? Like, how does this happen? Well, there's a lot of different mechanisms. On the whole, what ends up happening is you have enhanced depositing of cholesterol into cells of the artery wall, something called forward cholesterol transport. In large part, we think that's because macrophage receptors that are good at recognizing modified forms of LDL, for example, are upregulated. So SRA1, CD36, all these different scavenger receptors are upregulated on the macrophage. You also see a change in macrophage phenotype in terms of from a M2 to an M1-like phenotype. In the level of the liver, you see changes in LXR and FXR signaling and changes in bile acid synthesis and composition and transporters. The same thing happens at the level of the enterocyte in terms of changes in cholesterol absorption. And so just this compound TMAO putting into food or choline in the presence of a gut microbe to make TMA impacts sterol metabolism in multiple different compartments. And the net effect is to actually enhance not only forward cholesterol transport, but reduce reverse cholesterol transport. Now this picture on the right kind of illustrates how significant an effect this is. What this is are peritoneal lavage. The macrophages recovered from the peritoneal cavity of the apoe null mouse just on a diet with and without choline. Now there's no change in the cholesterol level in the blood. The LDL, the total cholesterol, the triglycerides are the same. And yet if you look at the macrophages, you can see this oil red O staining, how intense it is in the choline diet-fed animals compared to the normal chow-fed animals. So this is what an oil red O staining positive foam cell. It's the characteristic cellular hallmark of the atherosclerotic process. And so just changing the choline in the diet, and we see this only happens when it gets converted into TMAO, is changing how cholesterol is handled in the tissue. So it wasn't changing circulating lipoprotein levels, but it changed tissue cholesterol deposition. We'll get back to that in a little bit later. Well, actually, I put it here. So there have now been 3 different studies by our group and 2 others that have blocked this pathway by antagonizing or knocking down the FMO3, the liver enzyme, and with an antisense oligonucleotide. So looking at the top here, when you knock down FMO3, TMA goes up and you don't have as much TMAO. And the biggest effects we see are dramatic reductions in cholesterol ester in the liver. We also see changes in oxysterol and LXR signaling, but the bad side of it is you see enhanced inflammation, ER stress, and so we don't think it's going to be a reasonable target to hit this site in humans. But what we do see are very beneficial changes in terms of you get enhanced elimination of fecal sterol, more disposal of sterol, and also decreased cholesterol absorption. So by blocking the TMAO generation pathway at the level of the liver enzyme FMO3, you're eliminating more cholesterol from the body, you're lowering your absorption of cholesterol, you're changing your bile acid pool size and composition in a way that actually we think would be beneficial from an atherosclerosis standpoint. However, there are some downsides. The biggest being it's causing inflammation in the liver with this FMO3, and we think that's because there's other FMO3 substrates that we don't know what they are that are accumulating, just like TMA is accumulating, and this really stinks to high heavens. These mice are putrid smelling, and they're so bad that in a mouse colony where things smell in general, they forced us to move these mice into their own separate room. When you walk into this room, you feel little prickly stuff on your face, and it just hits you like a wall. The smell is so bad. And so people who have a genetic deficiency of FMO3 have what they call fish malodor syndrome, and they're social pariahs because they stink. And the only phenotype that's known with it is, besides the smell, is increased suicide. And it's unclear whether or not that's due directly to being like social pariahs or whether or not there may be an actual neurotransmitter function of TMA, because there is a little bit, although most of the receptors for it are in our nose, there's a little that you can detect some receptor in the brain as well. Oh, I shouldn't have done that. Let's see here. There we go. Okay, so I'm going to very quickly kind of run through some other studies that, by a variety of groups looking at different risk profiles for subjects. So this was done by a Canadian cohort where they were looking at a multi-ethnic cohort and saying what's the relationship between plasma TMAO and cardiovascular risk in this multi-ethnic Canadian cohort, and they saw an even steeper distribution between TMAO levels and cardiovascular disease in their cohort. Just recently published. A lot of this has all come out in the last year, so the field is finally starting to really pick up. It's not something you can just dial down to your local lab in the hospital to measure, so that's what's taken a little longer. Here's the EPIC Norfolk trial. We've served as the research arm, or the laboratory component. They're serving as the data coordinating center for it. They gave us the samples in a blinded way. We analyzed them in over 4,000 samples in a case control examination of, this is 28,000 community-based cohort who are followed longitudinally to see who went on and developed cardiovascular disease, kind of a supersized Framingham in Norfolk, England. And there's this is the odds ratio for risk for incident cardiovascular disease development in the 95% confidence interval. And you can see there's a fairly steep relationship that the higher the TMAO, the higher the risk for incident development of cardiovascular disease. It started with all the subjects we looked at had no known cardiovascular disease and middle-aged at initial evaluation. We've also looked at a cohort that we call like the COURAGE trial-like subjects. These are subjects who had significant evidence, angiographic evidence of cardiovascular disease, but the decision was to elect for medical rather than revascularization or medical therapy alone as opposed to in combination with revascularization. We are curious to see if in this higher risk cohort, if they had a high TMAO, would it help predict likelihood of having an adverse event in the future in this cohort? And it really, it's still a very strong predictor and largely independent of other traditional as well as renal and even heart failure markers, is a good prognostic indicator of future risk. Now, one of the things that we keep seeing in many of these cohorts is that TMAO is tracking with thrombotic event risk. And that's kind of illustrated here. This is looking at subjects that present to the emergency department with chest pain, and we just said, if we have a high TMAO, does it predict incident risk of an MI in that initial 24-hour window? Also, what about major adverse cardiac event in the next 30-day period? And TMAO is a very strong predictor of that. And in every cohort we're looking at, MPO is tracking with thrombotic event risk. So that led us to this question, is could there be a link between TMAO and platelet function? And so this is all new data that is now in press, should be coming out in the next 2 months. We began, actually, the first study we looked at was the original study we published in the New England Journal with TMAO, but we only looked at the thrombotic events and saw that even after adjusting for traditional risk factors, renal function, or the prevalent cardiovascular disease presence or not, there still was a strong prognostic value for the incident myocardial infarction or myocardial infarction and stroke. And this, in combination with seeing it in all these other cohorts, made us look at the platelet. So what we began with was, here's human platelet-rich plasma, or washed human platelets. We incubate them with plasma TMAO across the range of what we see in patient samples. All of these samples came from young, healthy individuals who were pre-screened and shown to have very low TMAO levels to begin with. And what we saw is that regardless of the agonist we used, whether it was thrombin or ADP, in the presence of TMAO, you shift the dose-response curve to the left and you make it more sensitive. So at a submaximal level of ADP, you get a higher extent of platelet activation, as indicated by aggregation, or a higher extent, at the submaximal level of thrombin. So here's the data with just 4 different subjects without and with TMAO, with ADP, or without and with TMAO with thrombin. But we've now actually, in the manuscript, we did over a dozen subjects with multiple different agonists and showed that every one of them, it shifts the dose-response curve to the right, suggesting a hyperreactive or a hypersensitizing the platelet. Now, mechanistically, as we started looking and trying to figure out where this was occurring, we were able to show that whether you used collagen or ADP or thrombin, each of these pathways were all affected equally the same, and it was above the level of inositol trisphosphate, because all 3 of these would not only shift the dose-response curve, but also generated more inositol trisphosphate as well. We also looked at the source of what was happening at the level inside the platelet. Why was it getting more hyper-responsive? And what we found is that it was impacting intracellular calcium signaling. So what we saw is that when we did Fura2-loaded platelets, we could actually demonstrate that resting plasma levels inside the cytosol of the platelet were unaffected. But in response to the agonist, the amount of calcium that was being liberated or released from intracellular stores in the ER was augmented. And the calcium was coming from the ER store, not the extracellular compartment. And so TMAO, and we think there's a receptor in the platelet, but we don't know where, is somehow activating in a very rapid fashion normal stimulus-dependent calcium signaling in the platelet, and now we're also examining this in multiple other cell types as well. Now, in vivo, evidence of an enhanced thrombosis potential is shown here. We put animals either on the chow or a high choline diet, so plasma TMAO here is in the range of the high end of what we're seeing in human subjects. We also labeled the platelets with a fluorescent tag, and what we're doing here is we're looking at the carotid artery by vital microscopy with a cut down and did a ferric chloride injury, and so we're looking at a thrombus as it's forming in the carotid artery. So it's an in vivo thrombosis model. And so we're visually seeing at the time zero is when you injure the artery with ferric chloride. And so the platelet clot begins, and the animals that are on the high choline diet or have the high TMAO, clot formation is faster. And then the time, if you look downstream from the clot, you can measure the time to cessation of blood flow as a quantitative measure of in vivo thrombosis. And I'm going to talk about that. Think of it as a bleeding time, but it's a complicated, in vivo thrombosis measure, time to cessation of blood flow, or occlusion time. And so a high choline diet is enhancing this. Part of the whole reason we started looking at this is the technicians in the lab were saying it was difficult to draw the blood from the animals on the high choline or the high TMAO diet because the blood keeps clotting inside the tube. When you're using a capillary or using a butterfly, and it literally clots faster. So here's data to look at the time to cessation of flow. So a shorter time is more prothrombotic. And if you put animals on a TMAO or a high choline diet, their TMAO level in the plasma is increased, and the time to cessation of flow is shorter, so they're more prothrombotic. This is conventional animals, so the animals on a choline diet make a lot of TMAO. In contrast, if you take a germ-free animal and you put them on a high choline diet, their TMAO level is virtually undetectable, and they no longer have the prothrombotic phenotype, but they do with the TMAO. And this effect we see is immediate. If we give an IV infusion of TMAO or an IP or parenteral TMAO and then immediately do the in vivo thrombosis assay, we see the effect within 5 minutes. I shouldn't say immediate. It's within the time of when we do the surgery and the cut down. So a 5-minute exposure is all that's necessary. Now, across all the different animals, if you actually look, we see that there's a very strong relationship between plasma TMAO levels and in vivo thrombosis potential. The higher the TMAO, the faster or the shorter the time to cessation of flow, the shorter the bleeding time. So the more prothrombotic. So this is true, we think, in animals. We've actually, in this paper, we do multiple different in vivo thrombosis assays and show that in each one, we see the same prothrombotic effect, and it's gut microbe dependent and TMAO dependent. It's choline dependent as long as the animal has an intact set of gut microbes. So the question is, how do we look at this in humans? And is this true in man? So what we did is the following experiment. We can take subjects and start giving them choline. You can go to your local grocery store and buy it on the shelf. Choline comes as a supplement. Lecithin it's claimed to amazingly fix all ails that you have, and so you can get lecithin for your bowels and even as a memory enhancer. It's amazing all the different advertisements we find. But anyway, the amount of choline that we gave in this was in a veggie cap was equivalent to 2 hard-boiled eggs twice a day. It's the egg yolk that has the phosphatidylcholine. And the subjects that we've enrolled in this study, we've now completed it. It's got 10 in each group. Were either omnivores or vegan. To be a vegan, they had no animal products for the preceding year. Now we'll focus just on the open bars, and those are the vegans. And I'm just going to point with this. So their TMAO level at the start is very low, 2 micromolar. This is about the bottom quartile level in our community-based cohort is about 2.4, 2.5 micromolar. After 1 month of supplementation, they're still staying on a vegan diet. Their TMAO level goes up tenfold. And the extent of aggregation that we detect here, this is a submaximal level of ADP. It's like 2 or 3 micromolar, I don't recall which, but it's a fixed submaximal level of ADP, and they have enhanced aggregation that's detectable. If we left them on the high choline diet for supplement for another month, we saw no additional change in their plasma TMAO and no change in their aggregation. Now, when we look at the omnivores, these are some healthy young students who are just generating some extra money by taking some capsules, and their TMAO level is low to start with. After 1 month, of choline supplementation, their TMAO went up 15-fold, and the extent of platelet aggregation also went up significantly. And then further staying on the diet, maybe a little bit increase, but not anything significant. So most of the effect happens, it appears, within the first 1 to 2 weeks, actually. Although in this study, we were only looking at baseline 1 month, 2 month. Now, you may now immediately say, what about if you're on antiplatelet agents? Well, we're kind of knocking them off one by one. We started with aspirin, and, I'm sorry, I had already, that was the omnivores or vegan and then the omnivore. So here's subjects that we did with aspirin supplementation. So first we'll talk through the open symbols. This is before they were on aspirin. So this is the extent of both aggregation is plotted in TMAO down below under control conditions without aspirin. These are actually healthy volunteers. They're all omnivores here. And their TMAO level went up again about 15-fold in the absence of aspirin. Then they, after a long washout period of being on the choline supplement, we brought them back, confirmed that their TMAO was back at baseline at the start, and then placed them on aspirin. And what was interesting is their platelet function is reduced. So ADP-dependent platelet aggregation at this level, you can detect the effect of aspirin. But then after being on the choline for 1 month, 2 months, by the time they reached the 2-month point, the benefit of the aspirin had totally disappeared. So the high choline diet overcame the effect of aspirin in this cohort. And it's a small study. It's about an N of 10 in each group. Or this is 7, but we've now gone up to 10. And so the bottom line is TMAO, high levels, we think are associated with a prothrombotic phenotype. Aspirin will help attenuate that, but they're both acting in the same pathway. So if you're on a baby aspirin and have a high TMAO, some of the benefit of the aspirin may be overcome. Now, this is only 81 milligrams of aspirin. Whether we can overcome it with more aspirin or what the effect of other antiplatelet agents, those are studies that we're in the process of doing right now. Now, in the humans, we've now taken all of our data and put plasma concentration of TMAO versus aggregation, and there's a pretty good correlation between TMAO and this measure of thrombosis potential in subjects. And so what I would suggest is that this pathway is linked not only to the development of atherosclerotic plaque, but also to a prothrombotic vulnerable phenotype, and perhaps this helps to link the association between TMAO and thrombotic event risk. Now, in very quick succession, actually I'm going to probably, in the interest of time, only do some of this so that there's time for questions. This pathway we think is also linked to the phenotypes of heart failure development and renal disease. The heart failure paper has just come out in this last, it's still in press, ahead of print. And so, but the clinical studies came out in the preceding 1 or 2 years. This is in collaboration with Wilson Tang. We looked at multiple different heart failure cohorts and TMAO levels. We saw that in heart failure, whether it be diastolic dysfunction or systolic dysfunction, that they have higher levels of TMAO than the non-heart failure. If you actually look at BNP versus TMAO by tertiles, I mean, it's really amazing. This group right here was healthy, stage 3 systolic heart failure that was stable, and yet a high TMAO and a high BNP in this cohort predicted over a 50% mortality rate. So actually it was this data that I was like, wow, how can you get 50% mortality in supposedly stable heart failure subjects? It made us really start looking at the animal model data. So this was the animal model. We looked at the TAC model. It's aortic banding. We put the animals on the diet before the banding to start with in this particular study. We put them on the control diet, the TMAO containing diet, or the high choline diet, and induced outflow obstruction, and then followed over time the rate of ventricular remodeling over time. So all the animals get TAC at baseline, and if you look at just the control, that's the chow animals, you have a decline in this, here's ejection fraction or shortening fraction. On the high choline diet, you accelerate the rate of ventricular remodeling and loss of function. The same data was seen on the high TMAO containing diet. We've also now done is in animals on antibiotics, we suppress the gut microbes, the choline effect disappears. If you actually look at the hearts on a high TMAO or a high choline diet, it gets larger, it gets actually heavier, and when you do echocardiography, the top is the plus or minus TMAO in the diet, the bottom is choline diet versus chow, and let's just focus on the patterns are the same. So on the top, if you look at the TMAO-fed animals, the hearts are heavier, the left atria are larger, and the lungs are heavier. They also have enhanced BNP, go up in the animals on the choline or the TMAO diet. When we look at fibrosis, this is looking in the ventricular tissue, the picrocereus red. This is a collagen stain in the ventricular tissue or around arterioles. And you see that on the TMAO or the choline diet that there's enhanced collagen. And we know now that this is linked to the TGF phosphosmad3 pathway. I'm going to skip through the renal disease just to give more time for question, other than to point out that if you measure baseline levels in subjects with renal function that's normal, this is the Framingham Heart Study Group, not done by us. They did an unbiased metabolomic survey and said what predicts future development of chronic kidney disease in humans in the Framingham cohort who had normal renal function and then go on to develop chronic kidney disease in the next 8 years. Out of the top hits, trimethylamine N oxide and choline show up as being significant in terms of predicting future development of CKD. And then what we found is in animal models, it induces the fibrosis and renal functional decline. The same kind of story. Here's the animals on the 3 diets, TMAO diet or the choline diet or the chow diet. And this is looking at collagen in the tissue. It's dose-dependently related. And it goes dose-dependently with TMAO level in terms of how much tubular interstitial fibrosis and loss of renal function you have. So this is the amount of fibrosis that occurs. When you look at the amount of loss of function or injury, KIM1 is an early injury marker, kidney injury marker 1. And again, the diet alone induced the kidney injury. It was a chronic diet exposure here, long-term feeding, but it leads to renal functional impairment, elevation in cystatin C as well. I said cystatin C back then. I meant BNP when I was doing the heart failure one. So BNP is induced by the heart failure, in the animals that we looked in the heart failure model. Cystatin C is induced in the animals we were looking at. They both are. I mean, we can look at both in the same animal models now. So I'm just going to end by saying that if we're trying to block this pathway, we can do it a lot of different ways. One way is the diet. And you guys are probably all familiar with the Predimed study with the Mediterranean diet. Well, if you look at the Mediterranean diet, the adherence to the Mediterranean diet tracks with lowering of TMAO level. This was reported by a different group. Also, omnivores versus vegetarian or vegans, there's a difference in the production of TMAO. This is urinary production of TMAO in the subjects. And we're in the midst of doing dietary intervention studies now. This had over 280 people, but it was cross-sectional, it was done after the fact. We're like prospectively randomizing people to diets and looking at this right now. And just in a kind of shameless, prelude to what I'm going to talk about later today, at the grand rounds, I'm going to talk about how we can make an inhibitor to the microbial enzyme step that makes TMA and is involved in making TMAO. And we just recently published the first paper on this in December of last year, that by blocking the microbial enzyme system that makes TMA and therefore TMAO, you can suppress TMAO levels in the animal and suppress diet-induced atherosclerosis. So our hope is in the future that we're going to be able to actually treat atherosclerosis or perhaps other diseases like heart failure, renal disease, thrombotic events by intervening on this pathway. And I think we need to think of statins as a Homo sapien enzyme inhibitor. There will be other microbial enzyme inhibitors that are not antibiotics, they're non-lethal, they're not killing the microbes, they're just blocking a pathway in the microbe, but influencing the host physiology. And I think this is going to be kind of a new approach to cardiometabolic diseases in the future, not just TMAO, but other potential pathways, once we figure out what the players are. And I'm just going to end, by giving you, what about this? So if you look at energy drinks, for example, This is actually drawn to scale, okay? This is how many porterhouse servings worth of steak, of carnitine, is in 3 of the tall boy Monster energy drinks. So they put carnitine in energy drinks because they think it enhances energy. But actually, when you look at the studies, the vast majority, there's like 1 that shows a benefit and 8 that show no effect. And yet, nonetheless, that's the thing that, that Monster, for example, includes in their energy drink, and many other energy drinks include carnitine, although I have to say that some of them have started to remove it from their energy drink. But no one has done long-term safety studies on these things. So if you're talking about something that can develop atherosclerosis over the course of decades, you'd never see it, and you wouldn't know whether or not there's, what the long-term safety is. And I'm not. It's a concern. Anyway, it's a moment. Okay. So at that, I'll just end and say that I want to point out some of the folks who did this work. Zheneng Wang did the original discovery work with TMAO. I mean, it's been a big team. But the platelet studies were done by Weifei Zhu and Xinmin Li. Wilson Tang's been a long-term collaborator on all of our clinical studies, and Jake Lucis and colleagues have been collaborators on the metagenomics analyses, and some of the heart failure studies were done in collaboration with David Leffer and colleagues as well. Thank you very much.
Stan, that was amazing. Thank you for a fabulous talk. Open it up for questions. I have a few questions, but start with the audience. Dr. Rogers.
Stan, thanks very much for coming and spending time with us. And I agree with Svati, that was a terrific talk. You spent a little bit of time talking about the TMAOs and how they may influence macrophages. But we know that atherosclerosis is a pro-inflammatory disease. Do you have any sense about what they're doing to some of the other components of the inflammatory cascade in the immune system?
The short answer is no. We haven't looked at other cell types, like what's happening to T cells or what's happening. We just haven't done that yet. We don't know. The only cells that we really focused on, we've been kind of ticking them off. So we focused on macrophage. Endothelial cell, we see a big effect in terms of activation, both in vivo and ex vivo in culture. And we've tried doing some stuff with some very minimal with myocyte, but really haven't hit that hard. Only a couple of little experiments. And then the platelet have, so I mean, it's early days. There's lots more to do. Yeah?
Picking up on that theme, could there be evidence that you alter genomic profiling of the interferon cascade, for example, with this intervention?
That's a great question, and in fact, there is evidence that we do. So in some of our studies, we have started doing like transcriptional arrays, and we do see both interferon and even STAT3 pathway influence changes. So, but there's a lot of changes that are happening. So, teasing it out, and what we've really been trying to do is rather than just describe, we've really been trying to focus on what is the receptor, what's the sensor. And it's been very hard to actually find. I mean, that's what we've been looking for. Is like, is there a G protein coupled receptor or something, or a protein that's known to regulate GPCRs that's coming out? And we have some candidates, but nothing that we can talk about yet that's what the signaling is. But what is clear is TMAO is having some very fundamental effects. The biggest effects we see are like in the liver in terms of how sterol and bile acids are synthesized and handled. You see very dramatic phenotypes there. And whether you're adding choline or TMAO to the diet, as we've done, or as other people have done by knocking down FMO3. And what partly complicates this is when you knock down FMO3, it's not just modulating TMAO, but you're also accumulating TMA and whatever other substrates the FMO3 interacts with, and yet it has very profound effects. So like in the LDL receptor knockout mouse, now 2 different groups have shown this. The hypercholesterolemia in that animal is completely eliminated by knocking down FMO3. So it goes from total cholesterol, and this is on a high-fat, high-cholesterol diet, and the 1,000 down to normal. And we don't know how that's happening yet. other than FMO3 is involved. And so we're trying to find what's the receptor or the signal that's doing it. So I think some of the phenotypes are going to be related not just to TMAO, but to TMA is the other thing. So one way to think about gut microbes, they serve at the interface between what we eat and how we experience that world. And so many, like you can think of TMA, for example, as a fed state hormone that indicates we've just eaten to our body because we just experienced a meal and it goes through the intestines via gut microbes. So I think, are going to be many compounds that gut microbes make that are going to work like the glucose-insulin axis in terms of fed and fasting state, in terms of regulating metabolism at the liver. I mean, think how much the liver is being showered by these compounds all the time. So we just have no idea what's going on there. Yeah.
Particularly interested in your CKD work, and I'm going to look the paper up, but do you have any idea what TMAO and the other compounds are doing in the kidney, and do you think the pathways are similar to the heart injury model?
We think there is overlap, but I don't know how similar it is. What we have been finding, and so it was a Circ research paper that came out in the beginning of 2015, and Tang is the first author. But what we saw is that chronic exposure to the high choline diet or to a high TMAO diet led to, over time, and it took many, many, many weeks, like months, led to renal functional decline and fibrosis. And when we started mapping out, we still don't know what the receptor of TMAO is, but we saw that phosphosmad3 and downstream signaling pathways were activated, and that was, we think, in part what was linked to the tubular interstitial fibrosis that's occurring.
I'll have a follow-up question related to that, and then I'll give it to Eric. Do you think the fibrosis is a pure fibrotic mechanism, or is it related to microemboli and lipid metabolism, and it's sort of a complex interplay between those? Unknown.
You're saying things I don't even know about yet. So BID, you said, BID metabolism?
No. You show that TMAO affects cholesterol metabolism,
Oh,
affects
yeah.
thrombotic pathways, platelet
Oh, I see.
reactivity. Do you think the fibrosis is an end product of those intermediate pathways, or is it direct fibrotic injury in some way?
We don't know. At this point, all we can say is in many different background strains of mice, if you feed them a high choline diet or a high TMAO diet, you will get eventually renal fibrosis. But if you put them on, if you suppress the gut microbes, you don't. And if you give them TMAO, they do. And the inhibitors, which I didn't talk about at this talk, can block that pathway by giving the high choline diet, but simultaneously with the inhibitor. We have a more potent inhibitor than the one we just published. That's the one that we're actually using in most of our studies. It's about a multiple-fold, a lot more potent, and it suppresses TMAO down to near zero, and you block diet-induced renal fibrosis. So I think that but we don't know at the cellular or the molecular level how that's happening yet.
So that was an incredible talk. I'll give you one specific and then one general question. The specific question has to do with what's the sort of half-life of TMAO in levels? Because it's somewhat striking to me. You'd think that people's diet would change, levels would jump up, which you showed, but that if you measured my TMA levels over time, they wouldn't be too constant. So I'm surprised there's such a good correlation with something that's a chronic disease.
So first, in terms of what the half-life of TMAO is, if you use a stable isotope tracer to look at that, you get a half-life in the 2 to 4 hour range. However, if you put someone and they fast them, their plasma TMAO just keeps on staying the same over a long period of time. And what that means to me is that they're just constantly making TMAO because the gut is a huge mucosal surface. There's a lot of bugs there in the colon, and you're just constantly a bioreactor releasing. We know it's gut microbe dependent, and if you just give a single dose of ciprofloxacin, the TMAO levels plummet just from the oral antibiotic, for example. So it's coming from the gut microbes, but it's not so hypervariable. Now, we do have a manuscript that we're in the midst of working on that's looking at the reproducibility of the TMAO level and looking at the inter-day and inter-month variability. It's actually, surprisingly, it's not higher. It's lower than triglyceride in glucose. It's higher than LDL cholesterol, but lower in terms of CV to triglyceride and in a metabolic syndrome cohort. and blood sugar. So it's in the same range of blood sugar, and I think actually blood sugar and triglyceride levels postprandially have bigger shifts than TMAO levels. So what really drives TMAO levels is the chronic exposure. So, because that just leads to permanent, within the time frame of weeks, elevated levels. Now, what we have done is, the question is if you change your diet, how quickly will it come off? So we have gotten samples and analyzed them from one feeding study where subjects were given 25% of their protein was from red meat versus 25% from white meat versus 25% from non-meat source. And it was all a metabolic kitchen, and they got all of their meals, and they were on each meal for a month, and then a washout month, and then another month. So a very expensive trial, because they got 3 meals a day for free for a long period of time. And when we look at that, what we see is that by the time they came off the red meat diet, on the red meat diet, their TMAO level went up, and on the white meat or the non-meat protein source, their TMAO level came down, but that's 8 weeks later. We don't know any, so within 8 weeks, you can achieve a significant reduction in TMAO. I just know that's the time frame for the ability to go down.
So very promising as I ate my steak lunch. But the final point, I guess I'll just ask you, is just what you're proposing, if you're right, which it looks amazingly provocative right now, is transformational for how we think about the disease. How have funding agencies and even your own colleagues, how accepting are they to this remarkably different paradigm?
Well, you get both sides of the spectrum. Some people are very excited by it, and others, understandably, are like, let's wait and see. And not all studies will always, even cholesterol, LDL cholesterol doesn't track with cardiac risk in every cohort we look at. So it's just time will say, and I hope you guys all go out and measure TMAO in every DCRI trial you guys run, and if it's right, which I think it is, it's gonna float to the top. And so, but I think that we're starting to, it's hard to know until time tells. But every cohort we have looked at has had a good signal in terms of TMAO tracking with risk. The signals that we're not looking at often are the community-based cohorts. Most of the ones are like what you guys have, the tertiary care referral groups, and there we're seeing, we're always seeing a bigger effect with thrombosis, with events, rather than cross-sectionally. So that's where I think that we really should be focusing on is, will it track with events? Because this platelet stuff, well, I was saying this morning, it looked so fundamental, I made sure that 3 different people in my lab did it and were seeing it, and then we went to another lab who's a platelet lab, and we gave them the TMAO and said, will you please do this? And so, and we've now have like 4 independent replicates around our institution before we submitted this thing, and it looks real every way we do it.
Great, thank you. So a reminder about cardiology grand rounds, where he'll elaborate more on this story and I think talk about some of the therapeutic implications. And thank you very much, Dr. Hazen, for a stupendous talk. That was really wonderful. Thanks.